Today, we're going to talk about the book that Koloman Rath and I have published a couple of weeks ago. The title is Viroids and Viruses, or Viroids and Virusoids, Nature's Own mRNA, Understanding Viroids, Hepatitis C, and Ending a Deadly Infection, which is the infection by hepatitis C. The two authors are shown in the back here. There is Kolo, and Kolo is on the talk and will be more than happy if there's a question to welcome you. I'm just going to start to read parts of this book. But before I begin that, why would I ever write, or why would we look at the viroids and virusoids? First of all, they cause disease, and they're in plants, animals, and in humans. They're alive as anything. They replicate, they hop from species to species, and they change slightly over time. There's a lot of them, and people don't really understand that. And now that messenger RNA is a drug, particularly with the COVID vaccines, now that CRISPR is a new gene therapy, now that CRISPR plus RNA are being used for cancer therapies, it's really important to understand what RNA can do. And here you have little pieces of RNA, and I'll read from the introduction of the book, little pieces of RNA, maybe the smallest living things, that can go from one species to another, cause disease. They have to transmit from one species to another. They have to get into the cell across a cell wall or a cell membrane. And as you know, if you know about trees or plants, cell walls of plants are much tougher to get across. They have to get across. They have to get inside the cytoplasm. They have to transit to the nucleus, where they go through a very elaborate replication process. Now, almost all of us who are interested in biology know that enzymes are proteins, but enzymes can be RNA. And these small little bits of RNA have enzymes that help them replicate. They cut the RNA and they splice it back together. All of them have that, except a very few that use cellular enzymes to do that. But basically, these little bits of RNA have a lot of information. Not only that, they are compatible with the mechanisms in the cell. RNA doesn't just drift into a nucleus, it is bound to proteins which carry it across. So these little bits of RNA are adapted to our cells in very interesting ways, things we can learn for vaccines and for other kinds of RNA drugs. Things don't just get into the nucleus, I go through a nuclear pore, which is very selective of what it lets in. As you know, all our cells have a nucleus and a cytoplasm, and these little RNAs adapt to that mechanism and get into the nucleus. Once they're in the nucleus, they do something else that's just totally remarkable, which we can learn a lot from messenger RNAs and drugs, which is they copy themselves. They have to have the right signals, so they recognize the cellular replication enzymes. All those are very complicated signals. And as one of the people listening, Roberto Petrarca, knows, one of the things we study is the interaction between proteins and RNA. It's really complicated. RNA folds up just as intricately as proteins do. And it's the shape as well as the content that matters to those exact interactions with the replication. Now, if that were remarkable enough, they do something very surprising. Normally, your nucleus uses the enzymes that's in the nucleus to take DNA and make more DNA. And that more DNA then makes the new cell. Well, that's a replication process which goes on all the time in our bodies. But these little bits of RNA subvert that. They capture the very enzymes, the polymerases, and some of them capture 3 different kinds of polymerase 1, 2, and 3. And capture it for their own ends to replicate their own RNA. So it's using a DNA directed DNA polymerase to take an RNA and make more RNA. Completely amazing that they can do that. If that weren't enough, they have to get out of the nucleus. Getting out of the nucleus is no trick. One of the things that I discovered when I was a professor at Harvard is how intricate the process is of getting RNA out of the nucleus. It's just as complicated as getting something in. There's special proteins, there's blocks all the way along the way. These little bits of RNA overcome all those blocks. Wouldn't that be useful for the new generation of drugs? If that weren't enough, some of them, the virusoids, make their own proteins. And they make them, since they're so small, they make them in a very interesting way. Sometimes they make multiple proteins from the same little bit of nucleic acid. Now, as you all know, the genetic code is written in threes. If you start it at position 1 and read straight through, you make one protein. You start at position 2, you make another protein. You start at position 3, you make another protein. Now, usually there's stop codons in starting with position 1 or position 2, but in this case, there aren't necessarily. So that's the first thing they can do. They can make multiple proteins from the same bit of nucleic acid. And if that weren't enough, some of them very likely use the reverse strand. So while you're making proteins going this way, you copy the RNA that way, and you can make another protein. And some of the most important viruses for humanity, like HIV, actually reads in the opposite direction. Roberto Petrarca and I are looking at that question for SARS-CoV-2 and some others. That is almost the end of the story. The RNA then has to transit out of the cytoplasm, across the cell membrane, across the cell wall, through the plant, and then get into another plant. All of that with a minimal of information. These are really worth looking at. We should study them. And I can tell you that almost all the people I know, in fact, had the CEOs of companies who are working on mRNA drugs, don't know that viroids even exist. Or if they do, it's really vague. They're not prepared to understand what you can do with a little bit of information. That's why this is an important book. It's important for today. It's important for people who want to think about the origin of life. It's important for people who understand DNA chemistry. It's important for people who want to know RNA enzymes. It's important as a tool to understand intracellular trafficking and DNA replication. Not to mention pathogenesis, how these things cause a disease that they do. This is really fascinating stuff. And I'm very fortunate that Koloman Rath has been with me on this journey to see what the latest is about this. And that's what this book does. It is an introduction written for people who are not necessarily specialists in molecular biology or enzymology. This is something everybody should read to know. If you're curious about life, if you want to know what the future of medicine is going to be, if you're actually a drug designer, you want to understand these. And I can tell you, we don't really understand how these are doing all the things they're doing. We have some inkling of it, and that inkling is given in this book. But I would say there's a good 10 or 20 or 30 years of work of some of our very best biologists to try to understand this and use that information to make a whole new generation of drugs. That's an introduction. And with that, I'm going to just read this introduction from the book. The book is available on Amazon. It's also available on my website, williamhaseltine.com. That's spelled with an S, by the way, williamhaseltine.com. And it's, I think, something that we should read. I'm going to read just first the introduction and then a forward. Why care about viroids and viruses? Viroids and virusoids are small circular infectious RNAs. Sometimes I'll say viruses when I mean virusoids. It's kind of a harder word to say. So viroids and virusoids are small circular RNAs. Think of a virus, but much simpler. All pathogens have their own tools of the trade. That's Scola's writing, which allow them to infect and transmit between different hosts. Of course, some pathogens have more complex toolkits than others. Viruses are quite minimalist in their approach, but viroids and virusoids are more extreme still. Where all viruses encode at least one protein in their genome, used to produce an enzyme that can replicate the virus genetic information, viroids and virusoids don't. Instead, they hijack enzymes found within the host cells rather than carrying their own tools. They simply make use of those already present on site. They are also much smaller than viruses. For comparison, SARS-CoV-2, something we all know now, is made up of nearly 30,000 nucleotides. The basic building blocks of RNA and DNA contain only between 220 of viroids anyway, and 450 nucleotides. A lot smaller. That's really small. Yet, okay, that's as small as they are. Virusoids are generally smaller than viruses, clocking in at about 1,700 nucleotides, still a lot less than most viruses. And so that's the first thing about them, is that they're small. There's a theory that maybe life before it was DNA was RNA, and these are left over. Or some people think that they are derivatives of RNA. And one of the things we're learning is how diverse RNA is. Roberto Petrarca and I are currently beginning to work on a book that describes all the new things we've learned about RNA. And I can tell you, from the time I was a student, he was a student, even 10 years ago, the complexity of what is actually happening inside of cells has grown enormously with respect to our understanding of RNA. And again, that's a reason to understand what virusoids are and what viroids are. They may have come from cells, or they may be the actual precursors of all living forms. There's a big debate in the literature, and it's going to take a long time to try to resolve that. Here's the actual introduction to the book. Flip back here a bit. Messenger RNA, as mRNA, has become a key therapeutic modality. Propelled into the spotlight by the COVID-19 pandemic, vaccines are only the tip of the iceberg. There's an mRNA revolution on the horizon. For cancer treatment, we're beginning to write about this. To gene therapy, to drug development, mRNA will soon be everywhere. And I really think it is the revolution of the future. It is our equivalent of the DNA revolution that's led to more than half of pharmaceutical drugs today and more than half of pharmaceutical sales. You don't just swallow a pill now. You get an injection, you get an infusion, there are proteins. All that is a result of the DNA revolution. Right now, we're in the first stages of the RNA revolution, and it's happening pretty fast. Some of these discoveries were made maybe 10 or 12 years ago, and already people are benefiting from the discoveries. It's like the DNA revolution. It was discovered how to manipulate DNA and read a sequence, et cetera. It was discovered in the 70s, and there were drugs being introduced into people in the 80s. That has led to this giant biotechnology revolution. There's an aspect of this which I think is very interesting, and that is RNA can be chemically made. You don't have to grow it up in large vats. You can actually synthesize it, like in very small amounts. So I think a room the size of a garage that can make 1 billion drugs a year. Well, there's a very important implication of that, and that's cost. Right now, all of these biotech drugs cost thousands, sometimes hundreds of thousands, sometimes even millions of dollars. I believe that in the future, using RNA to replace our protein, our antibodies, our gene therapy drugs it can all be done with injections. There's a real cost of manufacture is $10 or $20. And given the right circumstances, that can be a cost of these drugs, making it accessible to everybody in the world, not just the extraordinarily wealthy people who happen to live in San Francisco, New York, Houston, Miami, etc., which is what's happening today with many of these drugs. So the mRNA revolution is something really to pay attention to, not only for your own health, but for the health of the world. From cancer treatment to gene therapy to drug development, mRNA will soon be everywhere. At its core, the idea behind mRNA technology is a simple one. Messenger RNAs are transfected into cells and cause a useful phenotypic change. You put something in, you change the cell, you change the way the body behaves, or you make an antibody to what you've made, which is what the vaccines are. But before long, mRNA was thought as a potential therapeutic drug. Nature devised its own transmissible messenger RNAs, Viroids and Virusoids. That's this book. A study of how these small naked mRNAs can be, 1, transmitted from one organism to another, enter a target cell, 3, migrate from cytoplasm to the nucleus of cells, 4, redirect DNA-dependent RNA polymerases to transcribe the RNA, 5, in the case of hepatitis D virus, direct the synthesis of its own proteins, and C, transmit and transit from the nucleus and cytoplasm to exit the cell and restart the cycle of infection. All these are lessons we can learn to increase the efficiency of mRNA use as drugs. In addition to shedding light on potential futures of mRNA, this book also outlines the relevance of virusoids to the fatal human disease of hepatitis D and encourages us to do additional research on the topic. So that's the book. Now I'm going to describe several other aspects of viroids and virusoids. This is a picture of what a healthy potato looks like. Most of you understand that. And something that causes a major blight, which was really the source of virusoids. These virusoids could really stunt and cause all sorts of growth abnormalities. And to give you an idea of that, I have here, I think, a list of virusoids and viroids that infect animals. This is just a small picture. We are beginning to learn that these small organisms are much, much more prevalent than we thought. It's almost as if a whole invisible world has been revealed. We've taken the cover off and understood. And the way we do that is use these modern sequencing technologies to sequence hundreds of thousands of organisms. We put that data in the databases, then we use our powerful computers, think of chat or AI, to sort through it. Which ones of these are likely to have some similarity to something we know that's a viroid or virusoid? And when you do that, all of a sudden you find they're everywhere. They're not just what we know about. They're everywhere. Are they causing trouble? Sometimes they're probably peaceful parasites. And sometimes they're probably causing diseases in plants, animals, and us that we don't have an explanation for yet. So one of the biggest discoveries just in the last 10 years that we have all this sequence information, and we have these powerful computers, is how abundant viroids and virusoids are. Now, what is the difference between a viroid and a virusoid? It's whether it makes a protein. And there are some plants, like tobacco, that are infected by virusoids that make a protein, and that protein carries out some key functions for it. And us, we're infected by hepatitis D virusoid. Now, virusoids, I'm going to talk a little bit about that, but first let me talk about how abundant they are. So these things are everywhere. So let me just read from the book some of the plants that are infected. Potatoes, tomatoes, eggplants, citrus fruit, apples, peaches, pears, hemp, that's cannabis, that's marijuana. And that's going to be a big problem. It's a big problem for US marijuana manufacturers. Palm, which is a major product of oils. Half of Indonesia is devoted now to making palm oil, as well as Malaysia and some other countries, even avocados. So these, as an important source of food, are important pathogens. And we don't have very much to combat these. I would say we have no way really of combating it, except for hepatitis C, the D. And what we do is hepatitis D, these hepatitis B, you get around usually. And if you stop hepatitis B, you'll stop hepatitis D, unless you already have hepatitis B. And then you get infected with hepatitis C. That's another thing that can happen. Now, let's see if we can see what happens with animals and people. It turns out, as I showed you, first we thought it was just a few. Now we know that almost all animals have their viroids and virusoids. So that's the second thing, their ubiquity and their ability to cause serious economic harm. Okay, so now let's talk about a different topic. This book really talks about the fundamentals of viruses and virusoids, but it also talks about hepatitis D. Why should we worry about hepatitis B? Well, if you contract hepatitis B and it's not treated, you have about a 10% chance of dying, and you die of liver degeneration, fatty liver, and cancers. If you're unfortunate enough to get hepatitis D and hepatitis B, your chance of dying goes up to 50%. Half of those who get hepatitis D die, and they die pretty quickly. Now, hepatitis D is a little virusoid. We go into it in great detail, and we're still analyzing its genome. And we believe, certainly Roberta Petrarca and I believe, that there is an antisense protein that's also made, in addition to the couple of proteins it also makes. So people who have the infection of hepatitis B and hepatitis D can carry it. It actually gets packaged into the hepatitis B package and is moved around from person to person. Okay? And so if you stop by either a vaccine or you stop with a hepatitis B drug, you'll stop transmission. So it is, the good news is we can do something about it. However, the other thing that can happen is if you have hepatitis B and you encounter hepatitis D, that can get into you as well. Now, very recently, something quite disturbing has been discovered about hepatitis D. And that is, for many years, we thought hepatitis B could only be shepherded around from one person to another or within the body by hepatitis C. That means only those with hepatitis B could get it, and it was only a liver disease. All of a sudden, we've learned that hepatitis D is pretty nonspecific with what virus carries it around. Flu virus can carry it around. Other viruses can carry it around. And if it carries it around, it's going to get into those other cells, and it may cause a disease there. So all of a sudden, there is a new interest in hepatitis D, not only as a cause of serious liver disease, but maybe other diseases that currently don't have a definition. I'm talking about human disease now, that don't have a definition. What is a causal agent? Some of these diseases, especially the brain and other organisms, have no real etiology. We don't understand them yet. Maybe they're also caused by hepatitis D virusoid carried by common cold viruses, let's say SARS, even long COVID could be in part a result of carrying around hepatitis D viruses or other viroids or virus, especially virusoids throughout the body. So it's really worth looking at. Now, there is another hepatitis D, the C drug, that could stop hepatitis D all by itself. That's because one of the key things that happens is the hepatitis D virus has to get from the cytoplasm to the nucleus. And as I mentioned earlier, there's a carrier protein for that. that carries the information from the cytoplasm into the nucleus and across the cell membrane. And there are special carriers that the cell has for that. And some of the new drugs interfere with that carrier, therefore interfere with cellular processes important for hepatitis C life. And not only do they stop hepatitis C, they also stop hepatitis D all by itself. So there is some hope for that disease, but it is something that we really have to look at in considerable more detail. For those who are more biologically inclined, this book also has some very interesting questions about how polymerases are seen, how other proteins are seen. There's some really interesting details in the replication of these viruses and these viroids and virusoids that make it up. I'll just tell you my own interaction with this. I've been a virologist amongst everything for the last 50 years or more. I studied phage, I studied tumor viruses, I studied human leukemia viruses and AIDS. I've certainly become an expert on COVID in the recent past. But at various meetings, I would talk about the full range of organisms that are out there. And early on in the 70s, one of the guys who was there was Theodore Diener. He got the Nobel Prize, or if he didn't, I think he did actually, for discovering viroids, these potato spindle viruses. And all of his life, he worked to try to understand, are these left over from early life forms, or are these really derived, like many viruses, from animal and plant genes that have been rearranged and now get transmitted as viruses? And he never was able to resolve that question. But he went on to create a whole new field, which is currently exploding because of the DNA sequence revolution, because of the AI revolution, because of the mRNA use for vaccines, for treatment of cancer, and for gene therapy. So it's been a long story. It's been sort of a recurring motif throughout my life. And I've been privileged to be working with Kolo and the team that I have here in the US to write about these, to learn a lot more about this. There's a lot more to learn. Right now, if this were an iceberg, we not even know the tip that's above the surface. We might know 5% of what there is to learn about these fascinating organisms. But a good primer, and one that should be generally accessible, is this book. There's a lot to learn from these little things. The more you study, it's like studying a grain of sand. You can learn the whole universe and what a molecule is, what bonds are, what quantum mechanics is. You can learn how the universe is built from a grain of sand. Well, these are more or less the equivalent of a grain of sand in biology. There's a tremendous amount. And to integrate with our own bodies and cause diseases that they do, one thing we really didn't talk about is, how do these things cause disease? And I would say that it's an open question. We're just beginning to understand something called regulatory RNA, how the structure of our RNA can actually control which genes, which proteins, which RNAs are turned on and off. And sometimes there's a whole new set of drugs called silencing RNA drugs. Little bits of genetic information bind to and stick to things that are necessary for life, messenger RNAs, and enzymes chew it up. So there's a whole program called DICER, we talk about it here in this book, that some people think that copies of this RNA are chewed up into small little fragments that deliberately go out and knock out the RNA that is necessary to make an enzyme necessary for the plant to live. So it's called RNA interference, interfering RNAs, and by microRNAs, and by small RNAs, and by silencing RNAs. We're just at the beginning to understand that, and viroids are the key part of the key to do that. Also, some of these make proteins, and those proteins are known to mess up things as well. Other ones, like hepatitis D, can just simply outcompete. Remember, they're using the same enzymes that are used to copy cells. Well, some of these things are really efficient, and hepatitis D makes a huge amount of RNA of its own, and that means you can't make as much as your own RNA, and maybe you just outcompeted the ability to replicate cells, and that's another pathogenic mechanism. I think what we're going to uncover, and we're just again at the beginning of this, a whole world of pathogenesis, which will be important for understanding human and plant and animal diseases as well. We didn't talk about the importance of this for animal disease, but now that we know that these viroids are there, these virusoids are there, the question is, what disease are they causing our pets and our livestock? And what diseases are there in the natural world that may be coming to get us? We know the natural world is full of things that are adapted for their own niche. When they get into us, we're not protected and cause trouble. We've just gone through that with COVID. We may go through that again with viruses and viroids. Another reason to pay attention to it. Another reason to buy and to read my book. Our book, I should say. Koloman, my book on this topic. Thank you all very much.